This is 1-year-old Layla Richards, who was diagnosed with leukemia. Right now, she's currently in remission, all thanks to a gene editing therapy that allowed her to receive healthy immune cells. To do this, scientists didn't directly insert these modified immune cells. Rather, they inserted a modified gene from a healthy donor of T cells into Layla's own DNA. Layla's body simply didn't know the difference and expressed this DNA as usual, making healthy immune cells. As the saying goes, out with the old, in with the new. The main argument we will explore in this video is that gene editing can be used more ethically by setting a global standard, ensuring public trust, and differentiating responsible applications. So let's talk a little bit more about what gene editing really is. Your DNA gets transcribed into a protein, which does all the work in chemical reactions to show your traits, such as eye color, height, or hair. Proteins, however, can only be built from instructions in RNA format, which is another type of genetic material. Thus, DNA gets transcribed into RNA first. Gene editing adds, removes, or replaces DNA that codes for individual traits. So, you change your DNA, you change the trait. Our DNA is also really unique, so scientists can engineer a guide molecule that aligns with a target sequence of DNA, known as a guide RNA. The target molecule then marks this DNA to be cut by a DNA cutting enzyme known as an endonuclease. Once the defective DNA sequence is cut out, the body naturally repairs this segment, likely in a healthy way. One way DNA naturally repairs itself is by copying the code of its identical sister, its homologue. This process is known as homology-directed repair. According to the Genetic Society podcast, scientists can exploit the system by engineering a fake homologue that the DNA mistakes as its own sister, which it then uses to fix itself. So, as long as our engineering homologue has the changes we want, our new DNA will too. The most prominent tool in the gene editing industry right now is CRISPR-Cas9, which has all the components I've mentioned earlier, a guide RNA to locate our target sequence and an endonuclease to cut out this segment. CRISPR-Cas9 can be used in either germline therapies or somatic cell therapies. What's the difference? Well, germline therapy modifies reproductive cells, so its changes are passed down through generations. Somatic cell therapies target body cells, so its changes only impact its host. Now, this can cause some issues. We as a society like to be aware of what's happening to us. And if there's any change, we would like to be able to either agree or disagree to it. It is the right and duty to ensure informed consent. Unlike the barista this morning who gave me decaf without warning, like, how rude. Anyways, an embryo can't possibly give informed consent. So this raises the issue, how can we know our actions are approved by the person who's affected by them? Well, the best thing we can do as of now is to get the informed consent of legal guardians and overall societal consensus on ethical usage. This is exactly why setting a global standard is so important, which we will go over in depth shortly. Another issue is class inequalities. The standard baseline price for any gene therapy is tremendous as of right now, and only having these therapeutics available to those who can pay is unjust. As a result, this could increase the gap between classes in the aspect of healthcare. Finally, the topic of eugenics. Eugenics is the modification of certain traits across a population of humans, typically ones that are superficial, such as height, athletic abilities, and cosmetics, in order to create a more desirable next race of humans. People are considerably worried about what can happen if gene editing really gets out of hand. So, what are the proposed solutions? Well, let's dive deeper into those main arguments I mentioned earlier. Number one, it's important to not have only standardized national regulations, but global regulations that consider ethical questions such as these ones and more. According to Business Insider, the globe is already very divided by regulations, but standardized regulations would mean more streamlined approaches and education about the topic. Number two is gaining public trust, which can help clear any confusion between the domains of science and religion. Let's discuss this differing perspective about gene editing. According to the Pew Research Center, many Americans relate gene editing to nature, which can interfere with overall reliability for treating serious conditions. With more public trust, however, more funding can be invested in creating error-proof applications of CRISPR. This will lead to a more safe and responsible set of treatments involving gene editing. Public trust can also just help labs to pursue responsible research and can also reduce the base price of treatment, reducing class inequalities, like we mentioned earlier. Finally, number three, differentiating between therapeutic applications and superficial enhancements. As we talked about with eugenics, creating a desirable and uniform set of characteristics across all humans is problematic because it decreases the overall biodiversity of our species. This can make us all more susceptible to diseases and pathogens, and therefore it is not responsible use of gene editing. I believe to keep CRISPR as ethical as possible, gene editing should only be restricted to therapeutic applications. According to PhD Tanuka Biswas, CRISPR usage and gene editing in general have only increased in popularity since its first introduction in the late 1990s. So it's up to us to make sure that we use it in a responsible and just way, not only for our generation, but future ones too.